Let's start the second morning talk by Mario. Please, Mario.
All right. Well, let's turn this on, right? Okay. Better? Yeah. All right. Morning to all. Today, we're going, I think everybody's getting a bit more technical today, so I'm not going to be an exception. We have to go into some technical details. Of course, I am, let's say, scaling it to our objectives in this course. But what you're going to see today is something really, really important. It's really important for anyone who wants to know and understand evolutionary biology. Because what you're going to see is the language in which evolutionary hypotheses or hypotheses of evolutionary relationships are expressed. And although this is so basic and so fundamental to understanding all the rest, many people don't have a good grasp of this. Incredibly enough, even some biologists. Because this has not entered the curriculum completely. Like you see it here and there in undergraduate studies, and then people who like the subject go on to graduate studies and they understand the subject in more detail, and so on. But overall, the understanding of basic tree language is very deficient. Anywhere, not just here, anywhere. So what I'm going to show to you is like the essentials for understanding tree language, or how the language of evolution is expressed, or how you show evolutionary hypotheses. It's very simple, but this simplicity has to be grasped very firmly, like you know what it shows. So there won't be much history today, like yesterday, just a little bit so you know where this came from. Basically, what you're going to be talking today was initiated by this guy. He's a guy called Willy Hennig. He's a German entomologist. Let me see, there is something else here, yeah. Sorry about the noise. Who published this book, which is considered the ground zero for modern phylogenetic thinking. Why I say modern? Because this came, I think I mentioned this yesterday, almost 100 years after the origin of species. So it took a long time for real phylogenetic language, for a phylogenetic method to come about. It took a really long time. Because why? Because people thought that they already had one when they didn't. So this postponed the development of the field for many, many decades. So it's only when this guy came that things began to change. And interestingly enough, his first book on the subject was this one, Grundzüge einer Theorie der phylogenetischen Systematik, in German, 1950, published on something that resembles toilet paper. But for you to have an idea of the historical importance of this, a copy of this goes for $5,000 in the original one today. It's considered like a classic, like the edition was not very big. But at the time, nobody read it. First, because it's in German. Second, because it's not widely available. Third, because in 1950, Germany was not exactly endeared by the rest of the world, so it was not very welcome anyway. So it took some years until it was translated to English, 1966, that this work had its real impact. And basically what Hennig said, he was a specialist on flies, by the way. What Hennig said was that everybody has been getting it wrong in terms of how we express phylogenetic relationships. What we're doing is something that was inherited from pre-evolutionary traditions and is completely inadequate to express what we actually know, what we know nowadays about evolution. And he was very successful in his arguments. Of course, this was a complete paradigm shift, so it was not received with open arms. There was a big war that took almost 20 years to finish, so the paradigm was completely shifted. But it was shifted, it did, because today it's been, like, people don't even know how it was before. You teach that to undergraduates and they ask, well, how was it before? I mean, this looks like something that could have been developed 2,000 years ago. And indeed, it is a very logical, very simple system, and very much in agreement with what we know about evolution. Anyway, we see that tree thinking is not something new. Darwin already said, or expressed his view in terms of trees, and many people did. However, the trees have always been something a little bit subjective and weird. Like this tree is post-Darwinian. This is Haeckel. So it's late 19th century. So Haeckel was like the Stephen Jay Gould of the 19th century. He was very popular, he was very famous, he published a lot of books, he was also a scientist. And this is the kind of tree that he published. It looks really like a tree, and you see the names there, but you have a lot of difficulty knowing exactly what this means. I mean, it's very, very vague groups. Into the 20th century, we get things like this, which look a little bit more objective and technical, but in fact they're pretty much like the last tree we saw. Things didn't change too much. Like, what exactly is this? I mean, there is a group here. Why do you cut this line here? Why is this node here, not here? You know, there are lots of things that are just highly subjective and very difficult to reproduce and to quantify. And all the trees that you see that have this kind of shape, this kind of complication, are usually old trees. Of course, people try to rationalize what they're doing by inventing adaptive structural zones and things like that. So there's a lot of work on that. Really, if you look into it with critical eyes and not passionately, most of it is just bullshitting. You can't really be objective about it. So things only change with Hennig. Now, this is how trees look today, evolutionary trees look today, something like this. They're a lot simpler. Of course, you can draw them in different ways. The lines don't need to be like this. You know, like the square things, they could be just like this. It would be okay. But what matters is the information they convey. This is what is important. This is what I'll try to transmit to you today. So to start at the very beginning, we have to look at the parts of a tree. What are the parts of a tree? What's a tree telling us, a tree of this sort? By the way, a tree is also called a cladogram, okay? It's just the same thing. There is a technical difference, but nobody cares about it today because everybody talks about cladograms, so they just call it trees because it's shorter. That's it. So the tree is composed of, two things. The terminals, which are the actual entities from which you get data, which means specimens of living things. And then what comes below that. It can be sideways, no problem, but it comes below or to the left, which is basically a system of lines connecting these terminals. So what you see here when two lines cross, two or more lines cross, is called a node. It's in Portuguese, but I'll say the words in English too. A node. The translation is not exact. This would be a knot, but no, in English it's a node. And this is the root, which is the lowermost node from where everything else comes. Between nodes we have a branch. And this is pretty much it. These are the parts of a cladogram or a tree. What does this mean? This means a hypothetical ancestor. Why hypothetical? Because what you do when you build a tree like this is actually a hypothesis of relationship. You don't see these things happening. They happened a long time ago. Most phylogenies, I'm going to show you one which is actually experimental, but most phylogenies we have no idea, there are no witnesses. Evolutionary history is just too deep for any actual witness to have seen it. So everything that comes below terminals is a hypothesis of relationship. So you'll see later how this is done. But at this point, we're just going to see how we read it. Now, we have these hypotheses, and let's say we have this hypothesis ready. It's done, and you just have to read that. What does this mean exactly? When you see a diagram like this node, this hypothetical ancestor, means that these two species at the tip share an ancestor which is not an ancestor of anybody else in this diagram. That's all it means. In other words, these two are more closely related to each other than to any of the other elements in the problem, in your tree. They're closest relatives. Or in other words, you can say they are sister groups. That's the technical language. When two things come from the same node, you say they're sister groups, which means that they are closest relatives. So this one is the sister group of this one, and vice versa. Okay, if you ask why sister, not brother group, because just translated to English from German, and in German group is feminine, so it's Schwester group, daughter group. So in English there is no gender, so sister group, just a translation. In Portuguese, group is irmão, okay? In Portuguese, group is masculine. Just curiosity. There is no sister group and daughter group. and broader group that doesn't exist. Anyway, this is what a tree conveys. All the information in a tree is related to the position, the relative position in the hierarchy of these nodes. This is what conveys information. And this information is what organizes all biological information that we have. These hypotheses of relationships. So if you have groups like vertebrates, mammals, insects, and things like that, these groups correspond to nodes in a tree. These groups exist because they share an exclusive common ancestor. That's the only reason why a group exists. Before this tradition, groups were highly arbitrary. So people used groups like invertebrates, fish, reptiles, which are groups which are not real in a scheme of relationships. So it was very subjective. Only with this method, so this is quite new actually for something so basic. Biology has a very long history, and this is like 70 years old only, or less than that actually, 60. As a paradigm, about 40 years old. And it's such an essential part of the science. Anyway, this is the basis for groups and the basis for organizing biological information into evolution. Now, let's go on. These are just different kinds of trees, okay? So you just don't get scared when I show them to you in the next slides. A phylogeny can be seen as something, it's drawn differently here just to show, well, this is the actual phylogeny. That's how it happens if someone was looking at it. And then you can express it in a cladogram, but you can also express this tree in an additive tree that actually quantifies the evidence for each branch and expresses it in branch lengths. We'll see it's a little bit more. And then you have an ultrametric tree, which is something we'll go into more detail later. But first, let's go into more basic details about trees. Well, this is, for example, a tree of tetrapods. What are tetrapods? Are the vertebrates that have 4 limbs that walk on land. Okay, and modifications, of course. So here you have some fish, just as a outgroup, as they call it, and then you have amphibians, mammals, turtles, some reptiles, crocodiles, and birds. How do you forget about the colors at this point? I'll explain that later. But how we read this? We read this the following way: there is a group. What is called a monophyletic group. You can also say it's a natural group or simply a group because nobody uses any other kind of group today. That includes only crocodiles and birds. This is called the Archosauria. There is a bigger group that includes these two plus Lepidosauria, some reptiles, which is called Diapsida, and so on. So each node corresponds to a group. So each node is a hypothetical ancestor. Each node is a hypothesis of relationships. And you go on reading the cladogram like that. Sometimes you have splits in more than 2. That's no problem. It happens all the time. And you have sometimes names for these groups which correspond to the nodes. Sometimes you don't have names. You don't need to have names for everything. Okay, you can't have names for everything because there's too many species, too many nodes. But for some groups, you give names. Sometimes these names are very, very old, so everybody uses them. So we just continue to using them, but this time associated with a node. Notice that only groups that correspond to nodes are real groups. So they have names here. They are the only groups that actually exist. They have empirical existence. They exist independently of our ability to recognize them. Let's put it this way. If you have groups that do not correspond to nodes, for example, a group that's included mammals and birds, this has been proposed in the past because they have warm blood. Okay? This would not be a natural group because there is no node, no hypothetical ancestor that includes only these two. If birds and mammals were if birds were placed like here, then we would have a group, but they're not. They're far away. So this is not a natural group. This is what's called a polyphyletic group. Just because we have to give names to mistakes. That's the only reason to use these other names. Now, if we have a group that corresponds to the part in blue here, marked in blue, which is all reptilians except aves, except birds. This is also not a natural group. Although many people probably thought, I thought reptiles were a group. No, reptiles are not a group. If reptiles are an actual group, birds have to be included in it. Because birds are modified reptiles. So if you exclude birds, the rest of the group is incomplete. So it's not a group that corresponds to the evolution of this whole set of organisms. So the group marked in blue is also not natural. Finally, the group marked in yellow is natural because it corresponds to this node, which actually receives the name reptilian. If you guys are surprised about the position of crocodilia, this is not surprising to the biologists here, but certainly may be surprising for the astronomers. This is true. Crocodiles are actually the closest relatives of birds. Among living things, if you have fossils, you have many other things between the two, but among living things, these two are closest relatives. Okay? We can talk a little bit more about that if you guys are curious, but there are many characters supporting this. Crocodiles just look like reptiles on the outside. If we were blood parasites, we would think that they were really close, no doubt about it, because the blood proteins are just the same, indifferent from everybody else. So things like that give us the hint of relationships. It's not just one character, but many characters. All right. Huh?
How big a group are crocodiles?
Very small today, yeah. Only a few tens of species.
They don't even include alligators, right?
Oh yeah, alligators.
Yeah,
Yeah, alligators too. Yeah. Actually, it's interesting because everybody sees crocodiles as we see them today. You know, these sluggish things that look like lizards. If you go in the fossil record and look at primitive crocodiles, they become more and more dinosaur-like. The primitive crocodiles are very different. They're even herbivore crocodiles. It's kind of counterintuitive, but they were. Anyway, there are a few tricky things about trees that need to be made explicit. So I'm just going to turn on the lights here so I can write down. One of them is let me move this. For example, when you have a tree, the only thing that really matters is the relative position of the terminals of the species here. So this is a tree. If we change this tree to this, it's the same tree. Nothing changed. What this tree says, B and C form a group that does not include A, and A, B, C form a group. Two nodes, very simple. This says the same thing. B and C form a group, A, B, C form a group. So these two trees are exactly the same. There's no difference. You can change them. As would be the same, a tree like this. Also the same. So what really matters is the composition of each node. So this is the group ABC. This is the group BC. So this is all that matters in a tree. The resulting groups from the nodes, the hierarchy of groups. That's it. You can, other than that, you can move it as much as you want. So if you have a tree, just to get an example a bit more counterintuitive, if you have something like this, and something like this. Let's say A. Oh no, sorry. Oops. If we have a tree, these two trees, are they the same or are they different? The same. Because the groups are all the same. You have EF here, EF here, DEF here and here, C, D, EF here and there, and B and all others except A, and finally the entire group. So these trees are exactly the same. Consequence of this, the relative position at the tips doesn't matter. For example, here C is adjacent to A, is that right? Here C is far from A, D is in the middle. This doesn't matter. So the relative position at the tips don't mean anything. What means is the actual hierarchy of nodes. The groups are the same, the trees are the same, because the implied hypotheses of relationships are the same. So that's how you read a tree. You can't the most common mistake is to overread a tree. It doesn't really matter. If you have something like this, the branch lengths also do not mean much in terms of relationships. It means in terms of branch lengths, which is something different. But if you have something like this, A, B, C, and A, B, C, these two trees are also the same. The fact that these two branches here are very long relative to the middle one doesn't matter. These two trees are identical, B and C are closest relatives. That does not mean that A and B are closer together. Okay? So I insist, don't overread the tree. That's the most common mistake when people start looking at these things. An example of this, the cladogram we already saw in your talks. I think I also showed it, I'm going to show it anyway later in this presentation. Bacteria. Archaebacteria, eubacteria, eukaryotes. Eukaryotes have changed a lot. Archaebacteria and eubacteria, at least for the things we think are important, did not change that much. So our impression is that the two are very close, because they look similar, but actually archaebacteria are closer to eukaryotes than they are to bacteria, to eubacteria. That's why they're put in the same group, bacteria, for such a long time, because people didn't know about that. People didn't know enough about bacteria to be able to tell how different they are biochemically. So they're put inside bacteria. But actually one of them is a completely different lineage, and actually closer to us than to other things that look just like bacteria. Why? Because if you go to the common ancestor here, everybody looked like a bacteria. So what happens to these lineages when they split? They all look at more or less like the same thing, like bacteria. No? What changed was this branch here. So once this branch changed, these should look similar to us. They just look more similar because this one changed so much. This is a group that includes us, for example. So it was this kind of thing that messed up phylogenetic reconstruction for so long. People tend to think that similarity means proximity of evolutionary relationships, when it doesn't. The two things are different. Sometimes they agree, sometimes they don't. So what Hennig did was to separate the two things and bring order to phylogenetic reconstruction finally. So now we understand this very easily. If you go to the 70s, this was considered pretty absurd, just for you to have an idea. It was not, well, okay, it's true, but who wants to separate these two things that look so similar? Like birds with crocodiles, no, it doesn't make sense. When you have lizards that look so much like crocodiles. But now it does. Now it's when you look at a broader, more layers of biological information, it makes complete sense. Why? Because this kind of arrangement will reflect in many other characteristics, not just the ones that appeal to us as important similarities.
So is the basis for some of these things the genetic relationships, or you seem to imply that it's inclusive of genetic the DNA?
Yeah, everything. Yeah.
And other things as well, or
Yeah.
is it mostly the DNA?
Well, at this level, it's mostly DNA.
Okay.
At this level. Because they look so different, that's very
Right.
different to find any meaningful comparisons other than DNA. Yeah. Usually at these very deep levels of life divergence, you use DNA. Because if you want to compare a whale to a rotifer, I mean, what do you do? I mean, there's not much. You have to go to the genes, yes. But you can do it anywhere. The genes are important all over. Phenotype is, can get important in some levels. Okay. But you don't know beforehand where information is going to be, phylogenetic information. That's why we use all the data. Okay, so sometimes the historical information, phylogenetic information, is in morphology, when you don't find it in the genes, or they're difficult to find in the genes, let's put it this way. The two things have to go together, because we have lots of fossil information for which you don't have DNA. And fossils are very important to date things and calibrate molecular clocks. So it's good to have a good morphology still, because then you can get a lot of additional information. But the data, we're going to see a little bit more of that later. Right now we're just reading the tree. Let's see how we're going to build it pretty soon. Okay, so if this is well understood, all the rest is easy, because most of the mistakes in reading trees comes from over-interpreting things, like branch lengths and things like that. Of course, if you get this tree, and instead of having it this way, you put it this way, it's still the same tree. You see? Nothing changed. C and B still share a common ancestor, which is not the ancestor of A. So they are closest relatives. So it's still the same thing. We have to, actually, most of the work we had to do in the beginning, when this method became more common, was to dissipate old ideas about trees. No, you don't look at these things. This is not important. Yep?
I have a question. On last screen, you can see birds and crocodiles together, and the turtles are in the step before. So can I say that crocodiles and birds, you can't say which one is closer to turtles? That's my question.
Exactly. A good question. Yeah, let's go back to that slide to explain that.
Can you repeat?
Where is it? Yeah, I'll repeat the question. Let me just put this slide back here. Okay, he said the following thing. We have here birds, crocodiles, some reptiles. I think you forgot about this in your question, right?
Yes.
But okay, can I change turtles for
Yes.
Lepidosauria? No problem? Okay. He said, in this cladogram here, we know, okay, crocodiles and birds are closest relatives. But what about which one is closer to this taxon here? This is lizard-like things, reptiles in a restricted sense. None of them. They're both equally related to Lepidosauria. Why? Let's think about a cladogram now as a process that happens over time, the process of branching, okay? If time goes from here to here, that's common in biological diagrams. In physics, it's usually from left to right. Sometimes they are also, but it's very common to put the bottom to top. You have the exclusive common ancestor of all vertebrates here. Then they split in two lineages. One of them eventually gave rise to things we call fish, or some of the things we call fish. We'll see details on that soon. And the other gave rise to all of this. So when this split happened, these things at the tip did not yet exist, because they split later. So it doesn't make sense to ask, well, who is closer to this? This, which one of them? None of them, because it didn't exist when this thing differentiated from here. What existed was the ancestor of all of them. They are not yet individualized. That came later. So none of them is closer to this one than any of the others. Even this one. It's not closer. It may look similar as a byproduct of how evolution works. It's cumulative. Differences accumulate. But by any means, that's mandatory. Because you can't control the change in the other branches either. They come later. And they can do whatever they want. Once a branch splits, it's free to do whatever it wants. It doesn't depend on the other one. We reconstruct phylogeny based on detective work. Especially deep phylogeny requires a lot of work. There are many sophisticated techniques to detect the phylogenetic signal even when it is very, very, very weak. Even when characters that mean nothing outnumber the right ones 20-1. So, but this was developed more recently. Anyway, none of them. The answer is both. Both crocs and birds are equally related to Lepidosauria. Because when they split, they didn't yet exist. They existed later. They were the same thing. Okay? Does that make sense? Yeah. So that's how you read it. Okay? And this is very important. Because if you don't see it this way, you mess up all the consequences of having an evolutionary hypothesis. It becomes completely illogical. So you have to be strict about what you see in terms of the branching pattern. Okay? It becomes very natural. I mean, I've been teaching this to undergrads for a long time. And I tell you, it's easier to teach undergrads than teach graduate students 20 years ago. Because graduate students already had their mindset on how to read things. So you have first to clean up that mess, the mental mess, and then teach them how it was. When you teach people who never saw the subject, it was a lot easier. Because, that's how it is. Okay, pretty obvious. In 2 days, they understood everything. And they read trees perfectly. Of course, now everybody thinks like that. It's normal.
Sorry, what is the apsida? Is butterflies with them?
Which one?
The apsida.
Oh, it's the group that includes crocodiles, birds, and some reptiles. The so-called Lepidosauria. Reptiles without crocodiles and
The
turtles.
apsida is the name of the group.
Yes, it's the name we give to this group here.
Yeah.
Okay? So you can name these nodes. Because, I mean, systematics is very closely linked to classification. So classification means giving names to groups. So people give names. People like names to name groups. So if you want to refer to vertebrates, you use the name vertebrates to refer to all these things. Tetrapods. So these are just names that you give to nodes, okay? Just a labeling system. So the names themselves are not important. So I could call them any name I want. The reasons for these names being what they are just historical, because people have used them in the past. So you keep using them. Okay, they don't have any technical relationship with the rest. All right, going on. So this is what I just explained. You can rotate a cladogram as much as you want, and it is still the same tree. So all these three trees are the same, okay? Sometimes it's obvious, like in this case. Sometimes it's not that obvious, like in that case I drew there. Well, let's see, for example, this is another tree of vertebrates, but this time I'm putting more things here on the fish side of things. Notice that, well, we have mammals here. We are inside this group, okay? These branches, branches at the tip, they can mean not only species, but also groups of species. Okay, so this is fractal. You can split as much as you want. And inside a species, you can split it into individuals and go on forever. And in individuals, you can split it in genes. So this never ends. You can simplify it as much as you want, and you can split it as much as you want. What matters is that at the same scale, the hypothesis is the same. Like we saw here, this is the tree of life, right? Everything that lives is here. Eubacteria, archaebacteria, eukaryotes. I just simplified. Of course, I could split each of these branches in 1,000,000 tips. Then I have a very detailed tree. All depends on the level of detail you want, what the question you're interested on. Anyway, here is also a tree of vertebrates, but this time with more fish here. What do you conclude from this? Well, things that we call fish, sharks, ray fin fish, the coelacanth, lungfish, no. These things do not form a group. They don't have an exclusive common ancestor. To form a group, they would have to have a node that includes all of them, and only them, right? Of course, there are nodes that includes all of them, but they also include other things. This node here, for example, is the ancestor of ray fin fishes, and the coelacanth and the lungfish, but also of other things. So it doesn't count as it's not an exclusive ancestor for this group. So fishes, as we normally understand them, is not a group. Now, although you have departments of ichthyology and things like that, fish, technically speaking, is not a group. It doesn't exist. You don't use fish in classifications anymore because they don't correspond to an evolutionary unit. It may look a bit counterintuitive if you see it for the first time, but if you know a little bit more about the diversification of vertebrates, this is a more complete tree. That's just to help you out finding things here. This is where we are, tetrapodomorpha. We are one branch inside 40,000 things here. Okay, about 20,000. We are represented by this animal there. When you look at this, okay, you have echinoderms, starfish and things at the base, but after a certain while, everybody kind of looks like a fish, right? Fish, 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 not fish, 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 fish. Most vertebrates look like fish. We are fish. We're fish that got on land and became very different, but we are fish. In modern classifications, Osteichthyes includes us, includes tetrapods, includes mammals, includes everything, except the very basal ones, like sharks and things. These guys are really different. I mean, like lampreys and things like that. They don't even have jaws, things that every other vertebrate has. So they are really primitive. These divergences are very, very deep. And you gradually get closer and closer to things that look like us. But if you look at the whole diversity of the main branches of vertebrates, you see that vertebrates are fish. And they have a few highly specialized branches that had the silly idea of getting out of water. It became very different because the environment is very different. So we became very different because of that. But we're still fish. Deep inside we're fish. If we make our phylogenetic relationships, we're surrounded by fish. So what does fish mean? It means nothing. It means something. If we could say, well, what fish-like appearance is and see that as a characteristic, well, it would be somewhere here. And there are a few exceptions, like this guy and his friends, but most of the fish are here. So it doesn't mean anything. Fish is just an appearance of something. What you need is actually a tree that includes all the terminals and has the nodes, and you can talk objectively about groups and their time and sequence of divergence. Okay? So I just thought that showing this would make it more intuitive. Being a fish is just a primitive characteristic for a very large group that comes from a long time ago. Okay, sometimes, I don't know, it didn't appear. Oh, okay. Sometimes you can express a tree just by parentheses. I don't know why. This is silly. Let me finish this and then I'll just explain the whole thing. I forgot I had to put this animation here. It's probably for retards. Okay, you don't need that. Okay, sometimes you can express a tree just by a sequence of parentheses. This is more used in computer science because there are lots of, you see, this needs a lot of computing to be properly done. And this is usually how you express it when you input trees into a program. So if you see something like that, it's the same thing as this tree. All this means is that A and B form a group, and then C form a group with these two. Okay. You can express trees this way, so don't get surprised if you see something like that eventually. Now, coming back to an illustration from Henig's book. Of course, the hierarchy implied by a tree begs to be translated into a classification. And that's obviously what people did. They started translating trees into classifications of organisms, just like a set theoretical kind of thing. For example, you can also rank the nodes according to the Linnaean hierarchy, and that's how it was finally adapted to the Linnaean system of rankings, which is from this 18th century. We just continue to use this system of names because it's so old and there's so much information already attached to it. So if we change it drastically, we lose a lot of old information. So we keep using this kind of thing. But it can be adapted to modern phylogenetic thinking. Of course, we run out of ranks after a while because the tree is very big. I mean, in Linnaeus' time, there are not many things known. Now we know a lot more. So trees are huge, thousands and thousands of terminals, and this only includes a small fraction of the actual diversity. But we continue to use this because it's practical. In a certain way, it's practical to retrieve information. Okay? It's not very important. It's just something that stays there because it's practical. It has been used for too long to change easily. So this is, again, a phylogenetic tree of life. This is the same thing that I drew here before, without the names, just with the letters representing them. But here you have bacteria, here you have what looks like bacteria to our eyes, but it's not, it's actually a very different group. And finally, eukaryotes. We're here, animals. Okay? It's funny that people who work on this region of the tree of life, they tend to look at this like, oh yeah, these all look the same. We in a paramecium are more or less the same thing. We in an earthworm. For all phylogenetic purposes, they are really similar and really close. What matters is the differences here among these bacteria, which are really important. Anyway, this is just another way. More correct way of representing this would be this kind of thing. People are realizing that there's a lot of gene exchange in the beginning, in the divergence between these three major clades or groups. So there's a lot of horizontal transfer, as we call. They exchanged the genetic information. Eventually, they became more isolated. But that happens a lot in divergence of lineages in general. So this mess in the beginning is normal. As time goes by, it gets simpler. They stop exchanging so much. Well, sometimes you see a tree drawn like this, without a root. A tree without a root is not just something graphic. It actually has implications. A tree without a root is a tree lacking the temporal dimension. It doesn't have time. When you have a root, these are upside down, but it doesn't matter, you have a point of origin for this whole scheme. This adds the time dimension. You know that these divergences here happened before these divergences here. Okay? When you don't have a root, you can't order temporally. So you ask, well, why do you need that? Because computationally, this is a lot more easy, a lot easier, sorry. A lot simpler to do. Okay, that's the only reason. So there is a whole theory dedicated to the mathematics of unrooted networks. They're a lot simpler to deal with for computers. For our minds, it's not that simple, because you look here, you don't know where are the groups. You say, oh, C and D form a group. No, not necessarily. Depending on where the root goes, C and D may not form a group. Or let's see, alternatively, A and B, because the example is already here. A and B form a group, or it may form a group like it does here, but if the root is elsewhere, it doesn't. Like here it doesn't, and here it doesn't. So the tree is the root is very important to actually see the groups there. But graphically and computationally, the root is very simple. We just attach it later and order the whole thing, okay? So the real problem, the real tough job is reaching this. But this gets very technical. I won't go into these details at this point. Just that you need to know that a tree can be unrooted. And when it is unrooted, you can't see groups there. You can't overread this. Okay, the groups are not there yet. You need a root. Just to give you a more, sorry. And this has added sound. So in cladograms, you have a relative temporal dimension. What does that mean? It means that T is from here to here, or whichever direction you want to put it. But in essence, it means that this split, or the split of this ancestral species, occurred before this split, and before this one. And also that this one occurred before this one. That's all it means. It puts a temporal order there. When you don't have a root, you don't have the time dimension. Okay, so just for you to, this is just a matter of spatial reasoning. It's actually very easy. If you get that unrooted network and put the root right here in the middle, oh. There you go. That's the tree you get. Notice that the nodes still retain their relative positions to the terminals. Like node 2 is the node of origin of C and D, as it is here, and node 1, A and B, as it is here, okay? So that doesn't change. If you put the root there, oh, this is thankfully easier. If you put the root there, you get this tree, okay? Just, you get the root inserted here, then you have A, and then you have this node. Let me see if I have the, oh no, I didn't have, okay? Then you have this node here, and finally you have this node here. So the relative position of the nodes remains the same. And just to make sure nobody has any doubts about how this happens, is still if you put the root in some other spot, you have a different tree. So the root is important, but it's computationally simple. It is biologically complicated, but computationally simple. So people tend to take the tree away, resolve the problem, and then put the tree back. That's why we have unrooted networks. It was a very important tool developed in the 70s. So this tree was shown yesterday by Ted. It's a very famous tree of life. Over 2,000 terminals. You can't even, I mean, if I don't explain, you won't even know what this is. These are the names, as Ted showed yesterday. And where are we? Okay, we're here, somewhere here, right? Like you showed yesterday in more detail. This tree doesn't have a root. But what is this in the center? It looks like a CD, right? This is the center hole. This is where the tree probably is. We don't know exactly where it is, but it's probably around there. That's why they didn't represent the root, because nobody knows exactly where the root is. Why we don't? Because we don't have things outside of this to compare with. This is the only life we know. So people do not commit. They suspect that the root is at eubacteria, the ancestor joining eubacteria to the rest, but nobody can be absolutely sure. This is indirect evidence. Good evidence, but it's still indirect evidence. It's not the same evidence we have for the rest of the tree. This is different. That's why it's represented this way. It's a very interesting tree. And this is only a small fraction of what actually exists on this planet. I mean, biology, once it starts running, is very efficient at producing diversity. So there are millions and millions of species. This is the biggest one done at its time. It's not even a real analysis. This was just put together from several different trees. Yeah, so it's handicraft actually. Nobody can analyze that many things together. Nobody analyzed all that together yet. Okay, sure. Well, okay, we don't need more of that. You see, you can do phylogenetic analysis of anything. Well, but this is a very, very powerful tool. Once we have phylogenies, when phylogeny, actual phylogenies started in the 70s, the field exploded. Systematics was like this mushy thing that some people did in some hidden places in the 60s and 70s. So it's a very, let's say, downhill science. When phylogenetic came, it just exploded in terms of people doing it, in terms of papers published, journals, everything. It just became a science again. It gained new life. And the reason is, once you have a tree, you have an open door for many kinds of research which before were not possible. And you find many parallels that we didn't know before. It's absolutely extraordinary. For example, here we have a cladogram, a tree for lice. And here we have a tree for the organisms where these lice live. Because lice can be very specific to one species, as many parasites are. And we see that the two trees mostly agree. Why is that? Because the two things evolve together. So the structure you see in the host, the house of the mice, is the same of the mice, because where the host goes, the mice goes. And so if they split, the mice split. So there is an enormous degree of coincidence between the trees of parasites and the trees of their hosts. And there is only one explanation for this: the two evolved together. Okay? So there were isolating factors splitting the species of primates, for example. The lice went along with them, right? They're just living on them. So whichever factors resulted in these splits also affected the parasites. So this is quite extraordinary and gave rise to a whole new field called coevolution, studies of coevolution, because you can see this in several different levels. Also, phylogenies have been very important in epidemiology and things like that. Not just evolution, but specifically phylogenetic studies. I'm not going to go into detail of that. Just a case I find interesting is at one point, when the AIDS epidemic was at its peak, people wanted to know if a certain dentist had actually transmitted his particular type of virus to his patients. And like 1 million people went and basically said they caught AIDS from that guy, from contaminated instruments. And they actually had to do a phylogenetic analysis of the variants of the virus to discover which ones were infected by the dentist and which ones weren't. And they discovered it by this analysis. You see that some of them are away from the other group of patients. So they caught AIDS differently. But these guys caught AIDS from him. So it can be useful even in things like that. Once you have a very specific and testable method of phylogenetic reconstruction, it can become a very powerful tool for understanding history of living systems. And there are other practical applications as well. For example, if you guys know that there are many species of poisonous snakes. You can be bitten by any of them, but they're not, anti-venom for all of them specifically. So if you're bitten by a snake and you know that species, but you caught it, you take it to a hospital and they don't have a specific anti-venom to that one, you have to make a decision which anti-venom to use. Because the general one is not efficient enough in many cases. You have to be more specific because they're biochemically very different. So what you do, there are charts that tell you where that snake is in the phylogeny, in the evolution of the snakes, and then you choose the closest one you can find phylogenetically. For example, if you're bitten by this king brown snake, what do? You look where it is here. King brown, here it is. Okay, king brown is part of this group, and there is an anti-venom for one of this group. Then you use the anti-venom for that one. If you use the anti-venom for this one, it's not going to work as well, or not at all. You may die. So once you know which snake bit you, you just go to the cladogram of the snake, the tree of evolutionary relationships of the snake, and choose the one that is phylogenetically closer to that one. Unless you're lucky enough to be bitten by one that was the basis for the anti-venom. This is unlikely. There are thousands of species, so don't count on your luck for that one. You just have to go via phylogenetic relationships. And not just the regular classification, because not always it is phylogenetic. You have actually to go to the technical information there. trained people know how to do this. they identify this name. So let's get this one. So it's actually used this way. It's very efficient. Now, let's get a little bit into how we build phylogenies, how we build trees. How is it done? Because so far we have been seeing how we read trees. Now, how are they constructed? This is a very, very technical subject. It's actually the subject of this field called phylogenetic analysis, which is there are people who work on a little bit of that for their whole careers. Okay, so it's a very vast field. I'm just going to give you a general overview of how that is done, so you have an idea of how the logic of the procedure goes, and a few of the alternatives. The original Hennigian reasoning went like this. If you want to say that we are close to the great apes, okay, gorilla, chimp, orangutan, the gibbon, things like that, what kind of evidence would you use? You look at them, just from things we know. Let's say I tell you, yeah, we form a group with the chimp and the other big apes because we have hairs. They also have hair. You say, no, a dog has hair, right? A rat has hair, many other things have hair. So having hair is not evidence that these things share a common ancestor. What you want is characters that evolved with this possible ancestor and only in it, and then got inherited with this by the descendants, right? Now say, okay, let's say we have 5 fingers. That's why we are close together. Again, if you know a little more about anatomy, you also know that many mammals have 5 fingers. A turtle has 5 fingers, so that also doesn't mean much. But if I go and say, okay, we are close together because we don't have tails, I'd say, that's better. Why? Because most of the primates and most other mammals have tails. So this is possible evidence of something that evolved in the exclusive common ancestor of all these species and was inherited by them. So this is actual evidence that these things are related. It's not just some general things. Okay? So that's how the original reasoning by Hennig, he was not very quantitative at all, that came later. But that's how he thought about it. You need characters that evolved with the ancestors. And then these characters will tell you later how to build a tree. Because the problem is actually quite complicated. We're doing things we cannot directly test in terms of when they happened. Let's say you see phylogeny as a tree, an actual tree, a botanical tree, green with the trunk and everything, with the branches branching out. Our problem in practice is someone just cut the tree around across its leafy part. How do you call that? The treetop? Across the treetop. And look it down. So all you see are the section parts of the branches, right? The circles. That's all we see when we are trying to reconstruct phylogeny. We cannot go to the side and look at the tree. You have to look from the top and you see just those round things. Those round things represent organisms. So our problem is to reconstruct the original pattern of branching of the tree just by looking at these sections, at the evidence there. So it's not that simple. That has been developed over the years. So what Hennig did was a very ingenious method of looking for characters that evolved or potentially evolved with the ancestors and then reconstructing the phylogeny based on these particular characters. This is still used today in a very highly modified form, although there are other methods that have replaced it. So for example, what this illustration shows is, let's say we actually know the phylogeny is like this. This is just pedagogical. Okay, that's how it happened. At one point, after C was separated from the ancestor of A and B, something changed here. Okay, lost the table. Okay, some base of DNA changed. Can be anything inheritable. After this point, this thing got inherited by everybody. A split from B, and the same characteristic was inherited by both of them. So when you look back, you know that this is evidence of this, because the two share this. So what you want is to look for characters that evolved here at the base of the group. Because these are the characters that evolved when those things were only one, when they were together. When they split, this thing just got carried along with the branches, right? So what we are looking when we reconstruct phylogeny is the things that change when 2 or more things were one, and then they're inherited. That's why very short branches, let's say things split, and then Then 10 years later, split again, split again. So it gets very difficult to reconstruct phylogeny because there's not enough time for differences to accumulate. They all look the same. No? Very rapid speciation events usually result in a bush when you reconstruct the phylogeny. You can't resolve because there's not enough information. Okay? So that's how the traditional Hennigian reasoning proceeds. Okay. Notice, we have seen that in other examples, but I didn't put a name to it. Actually, the names are not very important. The logic of the thing is important. Let's say you have a similar phylogeny to the previous one, but this time we had another thing that changed on A only. What does that mean? This means that A became different for that characteristic from B and C. Automatically, B and C began to share something that they didn't before, just because A became different. So when you look just at similarities, you see, well, B and C are more similar. They share this character, the white thing. They didn't share this modification here. So this is the kind of mistake you have to avoid, because this does not imply that B and C share a common ancestor. And this happens all the time. It's just an expected consequence of the way things change in the branches of a phylogeny. So you need methods that allow you to tell this. Okay? Of course, in Hennig's times, the methods are indirect. His idea was, well, just look around more widely and you end up discovering how far that modification goes. And there you understand it. Today we have other means of doing it. Of course, sometimes evolution itself can deceive you. Convergence is a very common phenomenon in the history of life. Two things can look similar because they evolved the same thing twice. That's very common. We have this all the time. Things that go in the water tend to look like fish. Ichthyosaurs, cetaceans, dolphins, whales, and things like that, just because they function the same way, not because they inherited that. So this is also a very common phenomenon. I mean, the method has to deal with that. And it actually deals. Okay? For example, flying invertebrates. We know that flight evolved at least 3 times invertebrates, actual power of flight. And we know it's independent. They do not form a group. So flights evolved in bats and their relatives, they evolved in pterosaurs, and they evolved in birds. Flights evolved in these 3 times independent. How we know that? Because when you have a tree of vertebrates, they are far away from each other. So it cannot be a single ancestor with this particular characteristic. Of course, you can postulate, oh yeah, all tetrapods are flying in the beginning, and all the others lost it 1,000,000 times. No, that's very unlikely. We tend to minimize ad hoc hypotheses to explain the distribution of characters. Okay, there are two great ways of reconstructing phylogeny today. They coexist at war, but they coexist. One of them is called maximum likelihood, which is based on statistics, which is created by this guy, called Joe Felsenstein. And the other was created by this guy called James T. Ferris. They're both still alive and well and publishing a lot. And the two methods, they agree on the results 95% of the time. But they're very, very different in the way they work. Actually, the fact that they agree is actually a good thing. That means that they're actually getting close to some sort of truth. But they both have problems. I'm going to elaborate on that a little bit later. Let's first see the Ferris method. How it works. This time we're going to deal with, let's say, bases, DNA bases in a gene or something like that. Could be done for anything. Actually, I think there is another slide with a more palpable example. Let's say you have A, B, and C, and for a given position in a given gene, they have the following DNA bases. Okay, adenine, guanine, and things like that. What's the best tree to express that? According to the method called parsimony, which is the method by Ferris, you have to minimize the assumptions about transformation, which means that this tree is the best, because you presume that everything has G, and G changes to A. Okay? Could be the opposite, but the tree would be the same. Could be everybody A here and G here, no problem. But the other trees would require more change, so these trees are not as good, because they don't fit the data as well. You try to fit the tree to the data, okay? Just in terms of amount of change. You minimize change. the justification for that is just explanatory power. That's it. The tree that minimizes the step has greater explanatory power. So that's how you do it. You try to find the shortest tree for all the data. Of course, nobody does trees with one data, one character. It's thousands and thousands, so it gets very complicated at times. You can see it perhaps more clearly if we show just a phenotypic example. For example, if you have birds which is blue and two birds which are red, what would you say? Well, the two red birds evolved their redness independently here. Or would you say, well, they evolved once and inherited it, right? You'd prefer this one. This one is simpler. It just tries to make generalizations from the observations, and you prefer this tree. Of course, this is a very simplified example. It's never that simple, but that's how it works. You minimize the number of steps. While the other method, now looking at the other method, which is called maximum likelihood, is based on probabilities of change for each character. It was extremely laborious in terms of computation time, because you have to test every single hypothesis of change for every possible tree, for every node. So it was not widely implemented until rather recently. I would say from the 1990s on, it began to get really applied. But now it's very much applied, more than the other methods, although it's much more complicated. In this case, you have to have a model of evolution. There are many models. There are many more than these I'm showing. Some very simple. The probabilities of change between the bases are all the same. And they get progressively more complicated. And you can also have asymmetrical tables of probabilities and so on. But what you do is you try to find a tree that's called the maximum likelihood tree, that will maximize all these probabilities together. And then you have a maximum likelihood tree. So you see, it's a completely different method of phylogenetic inference. It's actually remarkable, as I said, that they often, or most often, reach the same results. This is a very difficult problem mathematically. You can see here how the problem grows in terms of possibilities you have to investigate when you try to find the best tree for a given set of data. If you have sequences, you can read terminals, okay? A number of species you're trying to solve. If you have 2, you have 1. If you have 3, you have 3. If you have 4, you have 15. If you have 5, you have 105. 6, 945, and so on. So this number is not growing exponentially. It's much more than exponentially. Actually, with many mathematicians here, probably guys will easily discover the progression. But when you get to 100, the total number of possibilities is larger than the total number of particles in the universe. So it can become a very complicated search indeed, because 100 is nothing. People do analysis of 1,000 nowadays. So it grows incredibly fast. The curve is like this. At the end, if you add one more terminal, it's like multiply the number of possibilities immensely. So it gets a very, very complicated problem computationally. This is actually one of the fields where people work today. How to do it, how to deal with this. This is part of the field called the super astronomical numbers in mathematics. There are mathematicians who work on this because there are not many practical problems that they can apply this. Numbers which have no physical correspondence. So it exists there in phylogenetic reconstruction. Anyway, I like this picture because I thought astronomers would relate a little bit to it. Of course, most of the data today is from DNA. What does DNA data mean? It became kind of a magical word for many people. Oh, DNA shows this, DNA shows that. Shows that, this is more related to this and that. Okay, but how is it done exactly? How do you look at a DNA molecule and reach conclusions about who is closer to whom? Well, of course, DNA is composed of many things, and one of them are these bases, which are actually, there are very few of them, only 4 in DNA, A, C, G, T. So all the information we take about evolutionary relationships based on DNA relies on these sequences. When you say DNA data, what we mean is actually a string of little letters, A, C, G, T. A, C, G, T, millions of times. That's how the genetic code is expressed in a living thing. And that's what we examine when we need to look at phylogenetic relationships. we try to assess the degree of similarity in these various things, strings of letters for particular genes. Notice that there are many, many, many. Nobody takes the whole genome to infer phylogenetic relationships yet, but we're getting there. Maybe 4 years, we'll be getting there. We're already trying, people are trying, but I mean, we can't trust it too much yet. But we'll get better in the next few years. I'm sorry, this is my watch. I had to leave it on because I need to see the time, sorry. Someone called, let me turn it off. You take care of the time for me. Okay.
15 minutes.
15 minutes to still have time for questions? Okay, so let's go. Okay, so what we actually have when we have DNA data, we have this kind of thing. This is just a little bit. Okay? This is immense because there are thousands of bases in any gene. It's normal to have 2,000 to 3,000 of these things in many, many terminals. So what we have is a table, which is called the data matrix, for the way we analyze it. And then we align the sequences because they're not all the same length. Notice that there are things here which are just gaps, because some sequences are longer than others. Some sequences are longer than others, and you have to align them to be able to analyze them. Okay, so what could this be? This could be this sequence got bigger, or the others got shorter. That's why we call it an indel. It could be an insertion or a deletion, depends on where you look. Okay. But that's what you see at the end. You see something aligned like this. Okay. Does this work? I mean, does phylogenetic work? Because this is the biggest problem. We do all this crap. You know, people working for years and years developing these methods. How, in the end, we know it works? It's actually, the results are reflecting actual phylogenetic history. How we know that? Some people thought that they could approach this experimentally. So they made artificial phylogenies to see how the methods would actually reconstruct. So they chose something that evolves very fast, like a bacteriophage virus that eats bacteria. You can make many generations in a few days, and then you can make your phylogeny, get the samples, the DNA samples from them at the end, and reconstruct phylogeny by various methods and see their relative performance in recovering the right phylogeny. And the results were very good. It's quite amazing. Most of the methods recovered it really well. So there is independent confirmation that the methods probably work. Is there more confirmation of that? Independent, other than, because many people say, well, one thing is how bases behave in viruses in 2 weeks, in 3 weeks, in 3 months. Another thing that can be very different is how the same sequences behave over millions of years in an actual, in an eukaryote or something like that. Yeah, it's a reasonable doubt. But there are other ways of changing. I'm not going to this is too technical to go now. I'll just, this is how they perform. The methods perform differently. Some methods do very well, and some methods do very poorly. This is a simulation. So for example, parsimony, if uncorrected, does not do very well. Neighbor joining, which is another method based on distance, doesn't do very well either. UPGMA even worse. But some others do very well. If you have enough, long enough sequences, which means big enough data, you end up converging to the right answer. So this tends to show, people interpret this as showing that the methods actually work. They work in simulations, they work on experimental phylogenetics, and they work in other levels as well. Wait a minute, which are the levels at this? I'm going to skip this too, maybe I'll approach this tomorrow, but now it's unnecessary. For example, sometimes you have the opportunity of checking your phylogeny against an actual geographical event. We know that things speciate when land moves, because animals and plants, they move with the land. So if the land moves away, the animals go with it and they speciate. So if you can date that event and you can compare that to the pattern you see in phylogeny, you have another door open for testing. So that's what many people do these days. For example, we know that there are frogs in India which are not related to Asia. Where'd they come from? Well, we know that India came from in Gondwana, and it was closer to Madagascar in Africa at some point, and then it was kicked towards Asia and hit Asia. actually raising the Himalayas there in the process. In this long journey, it acted as an arc. Lots of things on top of it. It's a big thing. India is big, so these things survived for all this time there, like on an island. When they hit Asia, they just got out of there. And these things are dated by independent methods. And this dating agrees in the cladogram with what you found before. Just looking at the animals, not looking at geology. So that's a third line of confirmation that these methods work. Okay, the dating, of course, is a much trickier subject. Why is that? Because if you try to look at divergence times, let me just show to you how this is done. You look at divergence times for certain organisms, doesn't matter which. As a lineage evolves, it accumulates change. Each little tick of this is a change. Let's put it this way. Okay, so as time passes, divergence increases. It becomes more and more different from its sister group, for example. After they split, this one also accumulates its own differences. So in the end, you have a summation of these differences. That means their distance, okay? This equals the distance I'll do it between these two. And this distance can be used to infer time. Why? Because in certain cases, we know that a certain gene suffers one mutation every million years, for example. So if you have 1, 2, 3, 4, 5, 6 mutations, you know 6 million years have elapsed since this thing splits from that one. And that's very interesting information because we can relate it to other things that also can be dated independently and see if they match. How do you calibrate these things? In some cases, genes behave in a clock-like manner. They are not subject to selection. They don't code anything. They just sit there. Large parts of the DNA is just sitting there doing nothing. So it doesn't suffer selection. It doesn't suffer anything. It just stays there suffering, undergoing mutation. So since these mutations are neutral, they occur at regular intervals. It's just like a chemical phenomenon. It has nothing to do with what the gene is doing because the gene is not doing anything. So if you have a means of dating this thing or actually refining this clock, you can then date many other things. And how you do it? You get a cladogram and insert things from the oldest known fossil. And you see that lineage is at least 60 million years old. And this gene has undergone so many mutations. So you just have a rate of change for that time. Okay? And this is a very powerful method because sister groups, when you have, like this example here, I think I mentioned it yesterday briefly. A is the closest relative of B. They are the same age because they had the same point of origin. So if this thing happened 10 million years ago, this lineage is 10 million years old, this one is also 10 million years old, necessarily. So archaebacteria and eukaryotes are the same age because they are sister groups, okay? They originate at the same point. But that's a little more powerful than that because based on this principle, you can extrapolate the fossil record. For example, let's say you know that B has fossils that are 10 million years old. And A, you have fossils that are 90 million years old. Okay, so basically that's more or less this way, right? But you can extrapolate, because the sister group of B has representatives from 90 million years old, you know that B also has to be 90 million years old. It's just a matter that the fossils are not yet found. Then you can extend this down. So if you do this to a whole cladogram, you can have a very complete picture of the times of divergence there. Okay, sister groups have the same age. You apply that over the tree, over the most complete fossil record you have, and then you have a rather complete timing of events there. Then, if you have lots of genes here, you can start calibrating these genes. Of course, the question I know you are thinking now is how you know if a gene behaves the same way here as it does here. We don't. We don't. You have to discover that. Yeah, because in the beginning, people thought when this was discovered for the first time, genes that were not subject to selection, people thought, wow, this is the holy grail of phylogeny. We don't need to do anything else. Just count the differences and the phylogeny is there. But then something called heterogeneity of of rates was discovered. So the same gene can behave very differently in the tip of a cladogram and in the other tip of the cladogram. So the rates are not always the same, they vary. The molecular clock is not calibrated in an absolute scale. It has to be calibrated locally. That's why big phylogenies are so complicated, because what the behavior of a gene you see, for example, among vertebrates, is very different from the behavior of the same gene in a millipede or a spider. So the rate of change is different. So I'm just showing you complications, okay? So for you to see, it's not that simple. We have to account for many different factors. So this rate of change made people think, no, you can't use the same gene for everything. So if you want to see how distant you are from a person from China, use mitochondrial genes, because they evolve very fast. So they accumulate differences that are informative at this level. But if you want to see how closely related you are to an earthworm, then you use ribosomal DNA, which evolves very, very slowly. And all this time, it will have evolved very little, and it will be informed at that level. So you use different genes for different regions of the phylogeny. Why is that? Well, one reason is obvious, because if it doesn't change, it doesn't have information, right? It has to change. But the other is a little less obvious. Remember that when you have two sequences, can you actually see what's happening here in this part? Or should I draw it maybe a little bigger? If you have two sequences that slowly diverge, they begin the same. Let's say C, C, T, G, G, A, T, T, A. Something like that. In the beginning, this is species 1. Species 2, when they were both the same, had exactly the same sequence. But gradually, it begins to diverge when they split, because mutations that happen here will not happen here, and vice versa. So eventually, they become different. And the difference goes up to a certain level. Let's say we have something like this, okay? A, T, A, A. Okay, so it's more or less similar, but with some differences. I see that there's a difference here, there's another difference here and here, another here. So there are some differences. We never know if the one changes this or this, except when the phylogeny is done. Okay, but it doesn't matter, because we're counting differences. But notice, as mutations begin to accumulate, you may have mutations that actually change this back to A. So you don't just count mutations. If you just do that, you underestimate the change, the distance, because the change does not follow the distance. In the beginning, if you have a graph of the divergence versus time, the curve is like this. In the beginning, the two sequences differentiate very much because the probability of having a back mutation is very low. But as time goes by, they begin to get full of change. And since there are only 4 letters, it's very likely that if it hits again, it may change to the original state. So gradually, divergence does not follow time. So you tend, depending on where you are in this curve, you tend to underestimate divergence. When we reach this level here, this region, you see that the sequences are saturated. They no longer carry any phylogenetic information because they basically were randomized by a mutation process. So you actually have equations that normalize this and make saturation tests for you to know where you are. So you can reconstruct this properly and use the right genes. There are many mistakes in the literature. When this began, people are over-enthusiastic. So you shouldn't trust molecular phylogenies that are older than 5 years or something like that, because usually they suffer from all these little problems. I mean, it's getting more and more sophisticated as time goes by. And it's a very, very powerful tool, but it's an ongoing process. It's still going. People say, oh, genome is going to solve everything. I guess my time is probably up, right? And people are probably hungry too. Huh? Okay. So let me just show you something. What happens? I'm going to show this tomorrow. This is pretty interesting. No. As you can see, I changed a lot along the way here. Okay, this is an example of how you see a genome when you get it. Because today you can get a whole genome, something made for you. You send it to Korea or some other place like that. They do it for very little money. And it's very reliable, it's very well done. And what you get back is a file with billions of sequences. Little ones, big ones, all mixed together. They don't assemble the genome for you. So this is basically useless information. because the genome is very, very, very long, and you have to put it all together in a straight line. How you do that? You get that file that basically is like 1 terabyte of letters, and the real work is assembling it all together. This is the difficult part. So when people say they're doing genomics today for phylogeny, don't trust it literally. They're doing chunks of the genome to do it together. Because what you have to do is what this picture says, is in the middle of the process, you're putting it together based on the agreements that you see. Some of them are long, some of them are short, and you use all the bits to finally reach a single string of letters. But to that you need a map. And the map varies according to species, according to groups. So you need a map of that group to help you in the process. So right now we have a shortage of people who is specialized in genomic informatics. And if someone wants to work on that and have a vast field ahead of him and a lot of work, because everybody wants, like a student who has talent on programming and genomics is like, ooh, no, that's what I need. Because there is a real shortage of people on that. Because genomic data are coming at a flood, and we simply don't have the means to analyze all this. But when it's done, it's going to be a very powerful tool, no doubt. That's when we really understand all the details. Okay, so I think I'll stop there. We can continue tomorrow. Thank you.